If half of your potential offspring are recombinants, that would mean the genes are unlinked. So then that brings the question of there being kind of a gray area. So I added this from last class because, the question is like, well, between 50% and 0%, there's a big range. So like, how do scientists like kind of determine that? The range that is helpful is like between 50 to 35%. If you're somewhere in there of the number of recombinants, we would say you're unlinked, okay? And then if you get somewhere below that, we say there's partial linkage, some degree of linkage. So whenever you're doing these the problems in the fruit fly lab, you're trying to figure out would these two genes be linked or not. If you drop below that 35% mark, that would be a sign that there's linkage happening, okay? Because you're never really going to get perfectly 50%. You might, but it's pretty difficult to. Okay. All right, so chi-square. I found some kind of notes on it. They're a little wordy, but the basic idea is like you're trying to figure out with chi-square. If you do an experiment, is the difference you see in your data, is that based on like actual differences that exist, or is it just chance? In other words, like you flip a coin 10 times and you get 6 heads and 4 tails. Is that proof that there is a 60% chance to get heads and a 40% chance to get tails? Or is it just, there was just chance? There's a small sample size. You can't conclude that there's actually a difference going on. That's kind of the idea of a chi-square analysis. So this is the formula for chi-square. You don't have to memorize this. This is on your AP exam formula sheet. The symbol for chi is X, like a capital X that's slanted, squared, and then this symbol seems really intimidating, but the summation of, I'll show you a little more like when we do a sample calculation how you use this. But basically, you'll have a bunch of different, you'll do this formula and you'll sum up, you'll do this plus another version of this plus another version of this, and you add up all these different calculations. Where the O is observed, E is expected, and then you divide by and square those, and then you divide by expected. So you have a bunch of observed, so when you do genetics problems, it'd be like observed numbers of like certain phenotypes minus the expected number of those phenotypes divided by squared divided by expected. So you have a bunch of different, you sum up all those different calculations. Okay, I'll show you a sample problem. If that seems really complicated, I promise you it's not, especially at your age. It's the math really isn't that hard. Now, what it can be a little goofy with chi-square is there is a null hypothesis. So a normal hypothesis in science is like you develop this like testable hypothesis that you're trying to prove with your experiment. A null hypothesis is a hypothesis you're trying to like disprove. So like the idea here is the null hypothesis is typically saying that there is no, what I was saying earlier with like flipping a coin. The null hypothesis says that any difference you see between your observed values and your expected values is just due to chance. That would be your null hypothesis. So anyway, so like if your null hypothesis is supported by analysis, you would say that any like difference you see between like your observed what you actually observe versus what you expected is just due to chance. Whereas if you reject the null hypothesis is not supported, then there is some reason why you had a difference between you observed and your expected. So that would be like in genetics problems, that would be like evidence that there would be like sex linkage, or that there was like the genes are linked on the same chromosomes. In other words, the null hypothesis for genetics problems is typically that there's like these genes are not linked. They are unlinked genes. They're doing the normal random segregation and independent assortment. Everything is just kind of going as it should. Whereas if you are not supporting the null hypothesis, then that would mean, again, that there's some sort of linkage happening. If that's a little confusing, we'll go through a sample problem and it should kind of clear up a little bit for you. Another thing with the chi-square is you're going to end up reading, when you do this calculation here, you get a number. And to know what to do with that number, you end up using this table, where to figure out what number on this table, they call it a critical value. To know what critical value you use on a chi-square table, you have to first calculate the degrees of freedom. So the degrees of freedom is the number of groups, and then for your genetics problems, we'll call it the number of your phenotypes minus 1. So in other words, if you have 4 possible phenotypes for like, if you're looking at a certain fly trait or like comparing 2 genes, you would minus that by 1 and you would have 3 degrees of freedom. And then for AP Bio, we focus on what's called a 95% confidence interval. And so I'll explain this like through the table. It's kind of hard to understand this through words. I'll show you the table and how you interpret it. Anyways, let's do a sample problem and then and it'll kind of bring everything together. So, kind of doing like, let's say we're doing a dihybrid fruit fly cross, kind of like what you're doing where you're looking at a double mutant, so black body and it's eyeless, crossed with a fly that is wild type for both traits. Thank you. Okay, and then you get an F1 that is all wild type, but it's going to be like a hybrid. It's going to be heterozygous for both of those genes. Okay, and then you're going to cross the F1s together, and when you do that, you end up getting this many wild types, so a whole bunch, 5,000 something wild types. You get this many normal body but eyeless, this many black body eyeless, and this many black body. So the question you're trying to answer here is, do these numbers, are these genes, the gene for body and for eyes, are they linked or are they unlinked? Is like the idea you're trying to figure out here, okay? And so using these numbers, these would be your observed. What you need to figure out is your expected, okay? Now, what you would expect, what your null hypothesis would say is that you should get a 9-3 to 3-1 ratio of phenotypes. Meaning, 9 out of every 16 flies you get should be the wild type, 3 out of 16 should be normal body and eyeless, 3 of 16 are black body and wild eyes, 1 of 16 are black body and eyeless. So what you need to do is you need to find your expected and then compare that to your observed. So how you would do that is, so you'd have a little table here. You would have your 4 different phenotypes. Here's the numbers we observed. And then this is your hypothesis. This would be your expected. Your expected here. And so you got to use your ratios, and basically you're going to multiply each of these ratios by your total number of offspring, by 10,009. So it would look something like this. The number like if you look at just the wild type, we said it was 9/16. 9 out of every 16 of them should be the wild type for both traits. So you would do 9/16 times the total number of offspring. So your expected value would be 5,634. So that would go right here. That's what you would expect to get for the wild phenotype. And we're going to compare that to what we observed and see, is that number, or is that statistically different than what we observed? Okay? And then you do that for all the other 3 traits. So for eyeless, you should have gotten 3 out of every 16. Multiply it by the total, you get that number, and so on. So then filling in our hypothesis, our expected values, this is what you would expect if the genes are unlinked, and we're comparing that to the actual data that we got. And so the question is, like, clearly the numbers are different, but like, is it statistically significantly different? That's kind of the question we're trying to answer with the chi-square. So how we would do that is you would use your chi-square formula. You're going to get your chi-square value. So how you do that is you're going to do, for each of your 4 phenotypes, that's why there's 4 bullet points here, you're going to do a calculation. So here your observed for that first phenotype was this number, 5,610, minus that, what we would expect, what we calculated with like what ideally you should get, like 9/16 of them, squared divided by your expected, you get that number. Do that for all of your 4 different phenotypes, and then you're going to add up each of those values, and this would be your chi-squared value of 0.3. The question is, well, what do I do with that? That's where we're going to use this table. Before we can interpret this table, we have to calculate the degrees of freedom. Because we had 4 phenotypes, you do the number of groups, number of phenotypes, minus 1. So you have 3 degrees of freedom. So kind of bring it all together. We're going to use, this is the actual AP exam formula sheet, what it would look like. We ran our chi-square test. We determined that you had 3 degrees of freedom. And on your, on like the formula sheet, you see a 0.05 value and a 0.01 value. For AP Bio, we use the 0.05 value. What this means is that we're 95% certain that the data we got here would be just like any differences you got would be determined by chance. Okay? So these numbers here, these are called the critical values. These are the critical values. So what we need to do is we need to compare our chi-square value, the number we got from using that formula, to one of these critical values. Now, to know which one you choose depends on which p-value you use, which for AP Bio is 0.05, and the degrees of freedom. So you find where they intersect, kind of like with the Punnett square. So 7.81, that is the critical value that we need to compare our number to. So to remind you, we got a chi-square of 0.3. So the question is, basically we got to figure out, well, how do I compare this number to this critical value? This is our critical value. Okay? So here's the idea. If this chi-squared value, if this number is clearly less than our critical value, it's less than the 7.81. So what we do with that is we fail to reject our null hypothesis, which basically means, so fail to reject, it's basically like, essentially means we're accepting our null hypothesis. In statistics, just kind of a, I don't know, my understanding is just sort of tradition. They don't say accept. I don't know. I don't really care. But basically, you're saying we're accepting the null hypothesis. Now, understand what that means. Like when you fail to reject, you're accepting the null hypothesis. What you're saying is that any difference we got in our numbers, so going back to these numbers, the differences between our observed and what we expected, this is just due to chance. Because our chi-squared value, when we did our calculation, comparing these two numbers, because this number we got is lower than our critical value that we got from the table, that means we fail to reject, meaning we accept our null hypothesis. So try to like, you don't really have to memorize this. Try to understand this intuitively. Let's say I got like 50 million. I get a gigantic, how do you even write 50 million? You get a gigantic number. It's not even possible, but say you get a number like that is numbers that are drastically different from you would expect. What that means is your chi-square value is going to keep going up and up and up to the point where you're going to get beyond the 7.81. At that point, you would reject the null hypothesis, and that would tell you there's something funny going on with your data. That there's something going on that's causing you to not have unlinked genes. That would be the idea. Okay, that's basically it. Let me see if there's anything else going on. So this first bullet point, this is just saying that when you would reject the null hypothesis would be when you get a value that's greater than that 7.815. That's the same as this number. Just the AP exam table they give you just isn't as precise. They don't give you as many digits. Okay. And then again, this critical value, this changes depending on the number of categories or phenotypes you would have during the genetic problems. Okay. That's the last slide. Is there any questions about that? I think a lot of it is just you need to do some practice problems with it. So on the quizzes at the very beginning of the slide deck, I gave you some practice problems there. But then you're doing these chi-square calculations at the end of the fruit fly lab. So that'd be kind of your practice for it.